Welcome. In this series, we talk with the scientists of the Dangoor Center for Personalized Medicine to get to know them a little bit better, to understand what's happening in their research laboratories, what developments they expect to reach the market, and what medicine will look like in the coming years. Today, we're fortunate to host Professor Jordan Chill, full professor as of a few weeks ago, from the chemistry department and the Dangoor Center for Personalized Medicine. Professor Chill completed his doctoral studies at the Weizmann Institute of Science in the Department of Structural Biology. He continued on to a postdoc at the U.S. National Institutes of Health, or the NIH, in one of the leading groups in the world, which focused on the application of nuclear magnetic resonance, or NMR, in studying the structure, dynamics, and function of proteins. Professor Chill.
Great to be here, thank you.
Thank you for joining us. So your field is structural biology, I'm told. What exactly is that?
Chemistry is the discipline of understanding how the composition of materials creates their properties. Mm-hmm. And simple examples of this could be table salt, which we know dissolves in water, or sugar. A more complex example could be maybe a plastic or chocolate, if you think of something more tasty. The question is, can we extend this idea and structurally characterize what is possibly the most complex system of all, and that could be the biological system. That could be the animal kingdom, that would be us. How does our body work on a structural level? And because it's so complex, it's really a question of, are the methods that we have today capable of addressing this question? And the answer is they are. And the field of structural biology, using a variety of methods, is the study of how structure makes biology work, in a sense, or to understand biology on a structural level. I kind of like to think of myself as a photographer who creeps into the system and takes a little snapshot of what's going on. Protein A meets protein B in a wonderful wedding, so to speak. All of biology, all of what goes on in our bodies is all the meetings of proteins with proteins, catalyzing reactions and causing things to happen. And if we can come in with that one picture that tells us what's happening on a structural level, that can be very, very valuable in really understanding how the system works. Any question you have about trying to intervene or interfere with these processes in the body, and of course this is exactly what we do in medicine, any such question will greatly benefit from having this picture, this roadmap of what's going on. So it's very, very valuable information, and the need for it is clear. The only question is, are we able to supply this type of information? And that's really what our research is about.
So, okay, so you're looking specifically at proteins. How do you look at them? You say a photographer, but what does that actually mean? I'm looking at you. You don't look like you're made up of proteins.
Right.
So well, what does that mean?
Right. Well, if you look on the nanoscale at proteins, they're really the mini machines that make biology work.
Okay. What does that mean?
Well, it means that enzymes, antibodies, receptors, channels, pumps, all these functions that go on millions of times in our body every day, every second, are all handled and mediated by proteins. Okay. And as we said, are we able to see the protein on this very nanoscopic level, if you want? And the field of spectroscopy in general is a field which involves using electromagnetic radiation to study the properties of materials. For example, the distances between atoms, how close they are, what angles they form between each other, and a collection of a lot of information, of many, many little pieces like that, can actually give us the information that we're looking for. And you have to add into that complementary biophysical methods, which in a variety of ways, which I couldn't even start to go into. Some of them are not my expertise, and it would take a long time. But anything that tells us something about where the proteins are located, one vis-a-vis another, their orientations, their directions, the distance between them, any information like that is contributing to this idea of understanding biology on a structural level.
So are you looking at specific proteins? You're looking at the distance between proteins? Because you threw out a bunch of different terms here. What are you, Professor Chill, looking at in your lab exactly?
Well, in research, you really need to focus. And even though I could think of 100 different interesting proteins to look at when I started my independent lab, you really have to choose a few because you need to get funding and you need to focus your research efforts on a given problem in order to have a chance to really make a dent and have an impact.
So proteins operate, different proteins act differently in your body.
Absolutely. As I said, the proteins have many, many roles.
Okay.
And just as a researcher, you need to, pick and choose. To some extent, you also need to choose the type of protein that your method is appropriate for. And we'll say a little bit more about that later, perhaps. So let me just get back to the proteins that you're asking about. So we try to choose proteins which are interesting on the basic science level because that is what we really do. However, it doesn't hurt to have at the end of the tunnel a little bit of an idea of an application. And therefore, we'll typically look at protein-protein interactions which are related to health and disease. And I put health and disease together because even when things work, you need to understand how they work. It's not only when they don't work, you need to figure it out. Even the homeostasis, what cells do all the time, is fabulous. It's wonderful that our body works this way. It's unbelievable that something so complex could really work this way. Examples are how do toxins that come from snakes and scorpions and spiders and even sea anemones interact with potassium channels in our cells. That's what happens when you get bitten by a snake. What it puts into your body blocks the channel with implications. We look at protein-protein interactions involved in cytoskeletal formation, which is important not only for specific disease, but actually also for cancer, because what cancer cells do when they try to invade blood vessels is they act to use changes in cytoskeleton to sort of get into the blood vessels, and that can also be studied using the methods that we use. And I have to admit that really any question that a biologist would have would be something I'll be interested in looking at. And as it turns out, we have collaborations with just about every university in Israel, and some I even need to turn down because we don't have the bandwidth to look at. But anyone who has a biological question and has learned something about the system and is still thinking about their proteins as red rectangle meets blue circle, which is what you see in a biology textbook, and wants to know a little bit more about how it actually works, we're the people to come to. And then with their knowledge and our deeper knowledge of structure, we can really get somewhere in a variety of types of questions. This works, by the way, also for questions which are more biomaterials. We look at peptides that form little bubbles of phase-separated liquid. And so it has all kinds of, we'll focus on things related to the body in this particular conversation, but it has many, many applications. And that's one of the things that I love about the method that we use, because it's central in chemistry, nuclear magnetic resonance, but it's been shown over the past 3 or 4 decades how it can also look at biological systems, and that's really cool. That is really cool.
So you've referenced a couple times the method, and it was part of your intro as well, that's what you were looking at in your postdoc using the NMR.
Yes.
So tell me a little bit about the NMR and specifically how it does relate to your research.
Right, so nuclear magnetic spectroscopy is a spectroscopic method that focuses on the nuclei, on the actual center of the atom, not the electrons, which makes it a bit unique. It works in the radio frequency region. That's the radiation that we apply. And the phenomenon was discovered in the 50s. This is 70, almost 80 years ago. And it used to be a chemist's tool. You would put a small molecule of 20, 30, 40 atoms into the spectrometer, which is a big magnet. And based on the signals you get, you would be able to analyze what the material is. Now, if I mentioned 30, 40 atoms, proteins tend to have 4,000, 5,000 atoms. So the mind-boggling question is it at all possible to even think of using this method of NMR to apply to such a large system? And when I did my first degree in chemistry, I used NMR for the just run-of-the-mill molecules, and I really, really liked that. Then for my PhD, I went to a group at Weizmann, Professor Jacob Anglister, who had learned how to apply these methods to larger systems, in this case proteins, but it could also be DNA and other biological molecules. We looked at proteins, And he mentored me through this process of learning how to apply the same basic idea, but on a much more sophisticated level, which includes inserting particular nuclei into positions that we want to see so that we can see them better, and developing new methods that allow you to look at these very large proteins but focus on a particular site that you're really interested in. All this in service of the method allowing you to look at very complicated molecules, in this case proteins, and being able to get this type of structural information, which at the very basic level is no different from the simple information you get for the molecules. It's just so much harder to get it. Now, I'm standing on the shoulders of giants who stood on the shoulders of giants. So our developments in the field, we've had some nice developments, but our expertise really isn't in breakthrough developments in the field. It's more to take the methods that are being developed in other places, put the little touch up on them that we want for our particular program, and then go along and use it to study the protein that we're interested in. But that is why I went to the NIH. Because at the NIH, there's actually a nucleus, no pun intended, of several NMR really world-class experts. And the person I did my PhD, my postdoctoral studies with, Dr. Adrian Bax, he's considered one of the forefathers of the field. So when I went to his lab, I really focused on learning the methodology, not just being a user, but being a bit of a developer. So now in my independent lab, I usually run relatively standard experiments, and the specialty is in the proteins we look at. But if we come along a problem which we don't have an answer to because the company hasn't written the software, we can jump in and do a little bit of that as well in order to really have all the tools that we need to apply to the project at hand.
So you mentioned a specific protein, right? You said every protein operates differently in the body, and then you gave a whole bunch of examples how people come to you with their questions and you can apply things, but I'm getting a sense that your lab focuses on a particular protein, yes?
We focus on a few specific proteins,
okay.
but that protein also originated from a collaboration with another lab. So we took that protein and went further with it after having worked together for 7 or 8 years, which were very fruitful, but then we sort of took it in another direction, which we're leading now. And it's really, of all the proteins we look at in the lab, in a way this is the one that relates best to personalized medicine. So I think it's appropriate to mention
Please.
it. The protein is called WIP, WIP, which stands for wasp interacting protein. It's a very large protein. It has, if you imagine a protein as beads on a string and each bead is an amino acid, this particular protein has over 500 amino acids, so it's very, very large. It's considered what we call a hub protein, in the meaning of hub as you would think of it today. Many, many other proteins interact with it. It's considered a multitasker. There are many, many interactions involved. It kind of recruits proteins from here and there in order to generate a cellular response. And its main activity is to control cytoskeleton. So our cells don't have a fixed skeleton. We don't have bones inside the cells. We don't have ligaments inside the cells. The cells are very fluid and very flexible. But there is what's called a cytoskeleton made of very fine filaments made from a protein called actin. And if a cell wants to move, for example, it moves by creating actin and rolling it along. That's how a cell moves. And that can be important, for example, in a cancer situation where you think you're invading into a blood vessel. That's really a cell on the move. So actin is involved there. So WIP has many, many roles, but it gets its name from the protein that it's most well known for binding to, and that's the WASP protein. We have said WASP interacting protein. And WASP is related to a disease known as Wiskott-Aldrich syndrome. Wiskott and Aldrich were two doctors separated by 20 years and the Atlantic Ocean. Wiskott in Germany, Aldrich in the US. But they came up with this discovery that in a family they saw a certain set of symptoms only in the boys, and it was clearly a genetic familial thing because a few brothers had the same thing. And it turned out that this disease is quite rare. It only occurs twice to 3 times every million births, so it's very, very rare. Wow,
that's very rare.
Very rare. And what it causes is a malfunction of these cytoskeletal activities. And what causes the malfunction is very interesting. The WIP is required to be like a chaperone or a babysitter to this other protein, WASP. It's supposed to bind to it and protect it so that it only becomes active when you want it to become active, and it only degrades and disappears when the body wants it to degrade and disappear.
So not with cancer, for example.
Cancer is the opposite situation where it becomes too active.
Exactly. It's not acting appropriately,
exactly.
if we will.
In this disease, what happens is that because something is wrong with the way the two proteins recognize each other, then the WASP protein, the one that's supposed to be chaperoned, can't really stay alive for long enough. The body just tends to degrade it very quickly, and then in the absence of that protein, things start to go wrong. In severe cases, we're talking about a lifespan of only 2 to 4 years, so death at infancy essentially, or early childhood. The mild cases cause thrombocytopenia, which is a lack of platelets or badly formed platelets, and other immunological issues that these boys will have. And those are actually the most interesting cases, the mild cases, because unfortunately, if it's a severe case, it's usually not a lot to do.
And they're not around very long.
Right, or the treatment that you try to, in order to alleviate this, you go for things that are very risky, like gene therapy or bone marrow transplants, and you feel you have nothing to lose.
Right.
But when you have someone with a mild case and they're already 12 or 13 or 14 years old, and they're doing more or less okay, do you subject them to this more, risky treatment, or do you try to manage it and hope for the best? And actually, that's the type of situation where we're trying to provide a little bit of insight in, because
you're working on manipulating this protein?
Not quite. Remember I said that we're the photographer?
Yeah.
So it came to our minds after working on this protein for many years and knowing the structure of how WIP binds WASP. All this is it's already behind us. We already know this. What really happens when there's a mistake in the gene and the protein is slightly malformed? What is actually happening? Are they not fitting properly? Are they separating entirely? Because that will be the first clue to what needs to be done in order to do something.
You need to diagnose where the problem is.
Right, but the diagnosis so far has always focused on blood count levels, red blood cells, white blood. It's all the clinical, but
not the chemical,
not the molecular aspect of the disease.
So you're looking at the molecular aspect of it.
We're looking at it. As far as I know, this is the first time someone is trying to look at this disease with this approach. And it works because nuclear magnetic resonance has this exquisite ability to really detect very small changes in the complex that pits WIP and WASP together. So we can figure out, is a certain part of WIP disappearing or leaving WASP and exposing it to this process that it's supposed to protect from. Our idea is to collect more and more mutations, and we know the mutations because any patient will, of course, go for a full genome sequencing, and they'll know what the problem is. So theoretically, one day we could say to parents, we know what's the problem in your particular case, and that's
very personalized
medicine.
Right.
So it could be that we would recommend a different therapy. I say therapy, we don't have any therapy yet in hand. We're very far from it. But as an idea, we would recommend a different course of action.
Something that would address it at a molecular level.
Exactly.
Right.
So, in theory, if we know where the problem is and there's a certain area where WIP is separating from WASP, why not bring a little surrogate molecule that can perform the job instead of WIP, and then perhaps
if that's the real issue.
If there's another issue, we need to do something else.
Right.
But again, the picture is worth 1,000 words.
Correct.
If you see the picture, if you have the roadmap, you're able to
They always say you need to figure out the problem before you can figure out the solution. Right. So you're taking a different perspective. Well,
seeing is believing. That's the motto. Seeing is if you see it, you can figure it out. And
Got it.
That's what
you're using the
biology,
technology.
Exactly. That's
but
what
you
it's about.
said also, this is, look, for the 2 out of the million, this is imperative and critical, and I don't mean to minimize that at all. As a parent, if it were my child, it doesn't matter if it's 1 million, it's my child.
That's
and so I'm not minimizing that. But you did say you collaborate with other laboratories on other things as well. So clearly this is not just about this particular disease you're looking at. What are some of the other implications or potential advances that will come about through the work that you're doing in collaboration with others?
Actually, I hinted to one of these in mentioning that the cytoskeletal process is also very important in cancer and metastasis.
Yes.
And actually, we have a very nice collaboration with another Bar-Ilan researcher, Professor Chava Gilchen, who's from the medical faculty. And it started out just like you described before. They had figured out the biology, but they didn't have the structural understanding. So they already had a molecule that when you injected it to mice, it stopped or it greatly inhibited the process of metastasis, which was amazing. And not to detract from that you need to have that. Then they said, okay, but why is it doing this?
Right.
What is the mechanism of action? Because usually when you go on a drug design project, you really want to have all the degrees of freedom you can, because you're going to need to change things to make this initial compound, to make it fit to various FDA regulations, various toxicity tests, stability tests, permeability tests. There are myriads of things you need to worry about, and you can't come and say, it has to be this molecule or else. Okay, so what am I allowed to change in this molecule? Can I take bead number 14 out of the 20 beads and change it, yes or no? The only way to get the answer to that is to see the structure, see where bead number 14 is, and figure out, is it an essential contributor to the binding, or is it something that we can dispose of and put in something else in order to help some other property of the molecule. So even before thinking of a drug design direction, it's very valuable to understand why it's working. We call this the mechanism of action, the MOA. Why is it working? How is it working? So even in that initial study, first of all, we got the peptide, the protein, we solved the structure, presented it to our collaborators, and then we said, let's run a test. Let's make a change that our structure predicts will absolutely annihilate the activity of this peptide. Let's make the change and see if our prediction is correct. So there is a correlation between what we predicted and what we got. There's a certain bead that if you take it out, we see a lot less activity. And we see that bead is important in the structure. Now what happens if you take that bead, which is a purple bead, just to continue the metaphor of the beads, and make it a deep purple to make it really...
Make it stronger
Make it
even.
stronger.
Right.
Then you get an increase of tenfold in activity. So now we have a list of all the properties, and we're now developing this further. We actually got support from the IIA, Rashut HaChadshanut, to continue this direction. And we have a grant which is supposed to bridge the academia to commercialization gap, because many drugs fall along the way. They look great in the paper, they look great in the bench, but then when you try to actually try them out as a drug, there are all kinds of problems and you don't get there. So we have funding for 2 years to play around with this and try to further improve the molecule that we have, testing it first at the magnet to see if it still binds, then going to in vitro testing, then going to mice, and hopefully one day into humans. And so this is actually a potential inhibitor of the metastasis of breast cancer in
the growth
of it. And that is absolutely critical, because in breast cancer, as in other types of cancer, it's not the tumor itself, but it's what happens afterwards. So if you can catch a tumor on time and stop the metastatic process, you'll be okay. Much, much better shape. So I'll say one more word, even more importantly, is that it turns out that when you treat with chemotherapy, somehow it even encourages the tumor to try and invade. So what this means is you really have to think about the invasiveness from day one, not wait for it to happen.
Because the treatment makes it go in other places in some ways.
You're trying to kill it. It tries to run away.
And it's trying to run away, exactly.
It makes sense from the tumor standpoint.
Correct.
And what this means is that it has the potential of not only being important in a metastatic situation, but anyone with cancer might need certain treatment of. So this makes it also, it might have a lot of impact.
Right. So the way you're describing it, now I understand from the beginning, you started with, you take a picture, but it's not just a picture. It's like the visual I have in my head is that you have researchers who are working here and they're seeing what's going on here, and you have almost like a microscope that goes down to the next level. They know these things work together, but they don't know why. You have to visualize it or get even lower down on the nano level, as you
Right.
were saying, to understand why something's happening at a certain level, because that allows you to manipulate it if necessary as you move through the process to strengthen it, to address
Exactly,
problems.
exactly.
It just gives a deeper kind of more molecular
Right. And if you come to our lab is divided into, as usual, there's a more wet part of the lab and a more part where the computers are and the students are working. And on the students' screens, you'll often see pictures of these proteins. So there are many programs that do this. This is nothing new, but to visualize the data that we've generated using the NMR, and then you look at the picture and you start to see things.
Right.
You can figure out why this problem is causing this issue, and once you see that, then you can think how to do something about it.
That's fascinating, and it's extraordinarily important, right? The why of things.
Right, the why of things.
The
is
why
very
of things.
important.
The what is important. You want to solve it, but if you don't understand the why, as
Right.
soon as something stops working, you don't know how to fix
Exactly.
it. So you are giving them the why, so that if something, it's great. It's great, like you said. It's imperative that they figure out how to stop it, but they don't know why.
Right. Then it's very hard to do that. And that explains why this is also important. Remember, you also have to make sure that we're able to do it. And,
Right.
and this is thanks to these developments. And I must mention, I always like to be fair. It shouldn't sound as though NMR is the only method that does this. NMR is one of a panel of methods. X-ray crystallography, cryo-EM, fluorescence methods, all these are all providing this same type of information. Each method has its strengths and weaknesses. Each method has situations where it's the most suitable to use to apply to a certain problem. And really the best types of studies come up when you collaborate, because
Right.
then you have all these methods working together.
Everything's bringing different information, and you
Exactly,
can combine
exactly.
it.
So it's not only collaboration with the biologists, it could also be a collaboration with other structural biologists, because our methods mesh together in a very, very
And give a
fruitful way
comprehensive
and give a
picture.
comprehensive view.
Right.
Exactly.
So it sounds like you're collaborating all over the place.
Oh, absolutely. and I have to admit, it's one of the things that I like the most about science: the ability to meet new people, learn new methods. A lot of it is abroad as well, so it's a chance to see how other labs work. It's always very very much of a learning experience. And it's also a lesson in humility. You're not the only one who knows how to do science. Other people do great science, and 1 + 1 sometimes equals 3, which is important.
Right? Socrates says the root of wisdom is knowing what you don't. How much you
Exactly,
don't know. I know nothing.
exactly,
That's the beginning of
absolutely.
everything, and that way you learn. Ezu chacham halomed mikol adam. Right? Who's
Precisely.
the smart man who learns from everyone.
Precisely, yes.
So all the wise people.
We're on the same wavelength on this, I see.
Right.
Yes.
Let's talk for a moment, though, how you got to your lab. You talked a few times. First of all, again, congratulations on becoming a full
Thank you.
professor at
Thank
Bar-Ilan. And you talked about when you started your lab. How did you end up coming back to Israel and to Bar-Ilan in particular? You were at the NIH, which is a very impressive place.
Right. The NIH is a fantastic place. Tens of thousands of people working on all these biological issues and questions. As I mentioned, I was in a very, very good group for the method that I wanted to study. And in general, just a wonderful environment. That being said, I think as a family, we set out to do the postdoc with the idea of we would try to return to Israel, and that would be where I would look to set up my own group. And looking around at the various universities at the time, when I came to visit, as de rigueur, you go to several institutions to check them all out. I have a little secret. I really knew Bar-Ilan first because my high school years were spent on campus. I studied at the school that used to be, it's no longer there. It now has its own beautiful campus, not too far from university. But I was on campus, and so I kind of knew Bar-Ilan. And I have to admit that it struck me as, I heard that it's very strong in the Judaics, and now is my time to come back as a scientist. I'm comparing the various universities. And I learned that at that time, when I returned, this is 2006 or so, Bar-Ilan was undergoing a major upheaval of the good sort, where many new experimentalists in the STEM studies would be coming onto campus. And it gave me the feeling that Bar-Ilan, even if it's not the horse that's currently leading in the race, it's the horse that's galloping fastest. And that means that looking forward, and now is that forward, right? Now,
Right,
50,
right.
16 years later, it gave me the feeling that this could be a good bandwagon to jump on.
We're going to follow the metaphor all the way
Right,
through.
all the way through, all the way through. But of course, it also comes down to nuts and bolts. Bar-Ilan was actually the only university that was willing to commit and purchase, this big magnet setup that I needed to do my research, which to the tune of $2 to $3 million. It's not only my system, the department used it, but it's a specialty magnet that's used for studying proteins. And they were the only ones who said, we'll do it, but if you come. Right. So that was, a real vote of confidence, and I got a very good feeling from the people I spoke to, the head of department, the dean, the VPR, the various people who were in place to give me, I guess, the reassurances I needed that, Bar-Ilan will be a good place to go to. I had a bit of a dilemma between going to Bar-Ilan and to Weizmann. Weizmann actually has this system already, so it wasn't an issue of buying it. But as I said, what I liked about Bar-Ilan was that it was kind of a chance to start another nucleus of NMR studies in a new place.
Right.
Not join into an existing operation, but
lead.
rather to lead, exactly. And I was lucky that at the same year I joined, another magnetic resonance researcher joined, so we were kind of the two of us together. And 4 years later, another magnetic researcher joined, so we're now a nucleus of 3. And I would say that Bar-Ilan is considered a leader now on the national level because of the number of PIs we've recruited in these fields and because of the instrumentation that we have, which up to now is second only to Weizmann's setup. We're really in a very good shape. 16 years take a toll on any system, and we're now in the process actually of trying to renew the system. We are lucky and happy and I think worthy of getting a Wolfson grant, which will allow us to hopefully, this is about a year ago, but it takes time to set up the system. So we're hoping that in about a year or 18 months from now, there will be a brand new magnet which will have many new capabilities, including, by the way, NMR is also an excellent method to study materials and in a setting of energy and sustainability. So we've sort of brought on additional abilities that NMR can provide to create a larger group of people who use it, because these Wolfson grants typically want to see,
right.
how will it benefit the community, not just a single researcher, and that's understandable. So we're trying to now construct a system which will be very versatile and have all these abilities. My hope is, and we're working towards this, is that the day is not far, we will be able to do NMR on a cell. So not take the protein out of the cell and look at it in our test tube, but rather take the protein that's already there and just look at the cell, which would mean this would be the most native as it gets. I mean, even taking the protein out is already, you could say, well, doesn't have all its friends or other protein friends that it should be playing with. You're taking it, you're looking at it on its own. So how does that work? And the answer is that is an issue. I mean, we can't do everything. But if we could do NMR in a cell by putting our protein into the cell,
you can see all the interactions
exactly.
and see everything.
So this system is supposed to have a component which greatly enhances the signal, so that even a very small amount of protein would be visible, and then we could follow the fate of a protein as it goes from organelle to organelle in the cell, from nucleus to membrane. This is all, it's a bit of a dream, but it is done in other places in the world, and where we want to join. We want to be there as well.
I believe you can do it. And I think Bar-Ilan's very lucky to have you. And both as a researcher, and I'll tell you, as somebody who knew very little about structural biology and was never a big fan of chemistry, you've made all of this seem really interesting, accessible, and important. So, b'hatzlacha as you move forward with this
very much.
Great new equipment and in everything you're doing. It really seems to be relevant in so many different fields. So
thank you for the chance to describe it, and it was a pleasure to discuss it with you, for sure.
Thank you. Thank you very much.